Okay, so 24.5 nucleic acids. Guide for reading. Key concepts. What are the functions of DNA and RNA? How long a base sequence of DNA is required to specify one amino acid in a peptide chain? What are gene mutations? What forms the basis for a method of identifying a person from biological samples? And what is recombinant DNA technology? Vocab. Nucleic acids, nucleotides, gene, and I'm not going to do the reading instruction. You can go.
Okay. Connecting to your world. Maybe people have told you that you have your mother's eyes or your father's nose. Although this is not literally true, your eyes and nose are your own, you do inherit the instructions from assembling the proteins of your body from your parents. In this section, you will learn that these instructions are called how they code your proteins.
Okay, so DNA and RNA. DNA stores the information needed to make proteins and governs the reproduction and growth of cells and new organisms. RNA has a key role in the transmission of the information stored in DNA and in the synthesis of proteins. Then on the next page, on 780, the genetic code, 3 bases of DNA arranged in a specific sequence are required to specify one amino acid in a peptide chain, or a peptide or protein chain. Next page. Gene mutations, substitutions, additions, or deletions of one or more nucleotides in the DNA molecule are called gene mutations. And 783, DNA fingerprinting. The variation in the DNA of individuals forms the basis for a method of identifying a person from samples from his or her hair, skin cells, or body fluid. And last one, recombinant DNA technology. Recombinant DNA technology consists of methods for cleaving a DNA chain, inserting a new piece of DNA into the gap created by the cleavage, and resealing the chain. Resealing.
Applications in medicine, applications in agriculture, and cloning. Back to Pitts.
Figure 24.14. This photo shows strands of DNA extracted from cellular material. Figure 24.15, the nucleotide monomers of DNA are linked together through their phosphate groups. Figure 24.16, the 2 DNA strands in a double helix are held together by many hydrogen bonds. There are 2 hydrogen bonds between each thymine and adenine, and 3 hydrogen bonds between each cytosine and guanine. Figure 24.17, the space-filling model shows only a tiny segment of a DNA molecule and gives you an idea of the enormous mass of the entire molecule. Figure 24.18, the abbreviated molecular structure of hemoglobin, the oxygen-transporting protein of blood, is shown here. A, the hemoglobin molecule consists of 4 peptide chains, each containing a heme group. B, in the genetic disorder sickle cell anemia, defective hemoglobin causes red blood cells to take on a distorted shape. C, normal red blood cells have a donut-like shape. Figure 24.19. The scientist is comparing DNA fingerprints. DNA fingerprinting is an important tool in the identification of people. A person's DNA fingerprint contains DNA fragments that are characteristic of his or her father and mother, but that would not be identical to those of the father or the mother.
784.
Figure 24.20. Included here are the elements of an experiment involving recombinant DNA. In this experiment, DNA from one organism is inserted into the DNA of a different organism. Figure 24.21. These genetically engineered tomatoes were altered to increase the length of time it takes them to ripen. And last one, figure 24.22. Dolly the sheep had no father. She was cloned from a single cell taken from her mother. Dolly died in February 2003.
Okay, so for your guys' notes, you're going to be, like we said before, but I'll say it again in case anyone missed it. You're drawing the DNA strand on page 779, and you're also doing the top part of the table on 781. And, yeah.
We'll give you a second to do that.
Describe, did you have for the test? Did you have?
Yes.
You can just talk about it.
Okay, so for the test, we have, in this section, it's, doesn't matter what questions they are, but the questions that are on the test are about the, put notes on the other ones. first one I didn't. Okay, so it's about the DNA and what proteins do in the DNA and RNA sequence. And that is that they store the information needed to make proteins and govern the reproduction and growth of the cells and new organisms. And RNA has a key role in the transmission in the information stored in DNA and in the synthesis of proteins. And yeah. Let's see baby's back. Okay, so a typical nucleotide is composed of a base, a phosphate, and a sugar. And the base is usually, how does all my Okay, the base is usually nitrogen, and that's just important to know, I guess. For this. And base pairing in DNA involves hydrogen bonds, and they help to make DNA. So, okay. Oh, why do A and T and G and C pair up like they do? Like, because A and T is always together with G and C, and they bond because the hydrogen bonds like connect together because they all have certain ones that, like, for some reason, like to join a particular other letter. And so this was the weirdest one trying to research to be honest. So termination codes are there's three of them, three words in. I don't know why. Honestly, I do know that they're, like, in most science and chemistry, they teach them as nonsense codes because they don't ever make any sense, but they're words. But I don't understand what makes them termination codes, because they're just the codes in, like, the 3-letter codes in the chart that you guys are drawing. And there's a couple more. The technique of identifying a person is DNA fingerprinting, and it can identify someone with just about nothing, like a small, like, molecule of their anything. It can be like their eyelash or a strand of hair. It's pretty cool. And we only have one more question from this section, and that is approximately what percentage of a human's DNA is used for coding information for proteins. And I remember Rasmussen saying that that's pretty much the 5% that is most of what we know. Like there's still a lot more about DNA and sequencing that we don't really understand.
Okay, so everyone's done. So turn to page 785.
And
okay. So we're doing. I have to say the thing, don't I?
Write down section 24.5 RQ 1 and 2, page 785.
785.
2 and 3. It's 2 and 3. We changed the 1 and 2 to 2 and 3 because number 1's too short. Or too long.
It's too long. We decided.
If you already did it,
that
that's cool too.
It's RQ 2 and 3. You don't have to change anything. You just write 2 and 3. You don't have to, like, you can write 1 and 2 if you want to, but.
Oh, okay.
Because these aren't even the right numbers. It's 20, 27 and 28, but we go by, the section. So
We just try to make it easy on you, but if you guys already jumped ahead and already did it anyways, then we can do that too.
Okay. Nobody does that.
So who wants to read number 2?
Yeah, Rylan.
Just the question.
What does a 3-letter base sequence of DNA specify?
Capital T, 3 bases of DNA arranged in a specific sequence.
Wait, what? 3?
3 bases of DNA arranged in a specific sequence are required to specify 1 amino acid in a peptide or peptide chain.
It's at the bottom of page 780.
Okay, thank you.
Yep.
Oh, there it is.
Woo.
remember, hydrogen bonds are as an example, the hydrogen end of a water molecule being attracted to the oxygen end of another water
Look on your
molecule.
face and so many
It's
words.
a electromagnetic, it's not covalent, it's not ionic, it's an electromagnetic bond. And these nitrogen-based pairs, they have no hydrogen in them at all. But they do have the negative and positive charges that we identify with hydrogen bonds that are holding them together. And they come apart easy. They're not near as strong a bonds as covalent, and really not anywhere close to ionic bonds. Those are the solid ones. But that's why we can unzip it so easily to do that transcription. Just undo the middle of the DNA molecule because it's all hydrogen bonded. Yeah.
You know, when I graduate, I'm kind of
So
going to
number
be rambling on
2
about stuff.
says.
Really?
Yeah.
Do you follow it or?
Maybe that's my
Number
number
three,
3, not
but it's-
What are the
graduate.
3 types of
So we are moving on.
Okay,
mutations?
we still have to do 2, and then we're done.
I know.
I don't
we
know,
have
I
to
just
do
get amazed by this stuff
trying to-
sometimes. I hope I transfer some of that
Yep.
amazement.
I wasn't done with
It
it.
is on
Let's be
page
efficient. What do we have to do?
7782.
Don't ramble.
Yep. Chompy chompy.
Yep, that's all you need. Okay, I'm gonna go right now my vocab.
Chomp.
Are you done?
Yeah.
Let's hear it for 'em.